Professor Said Gezgin from Elemental Analysis Lab. Two professors, classical breeders today with us, and Professor Kampoch will give another talk related to actually directly the utilization of the knowledge and genes related directly to salinity tolerance. And Professor Topal is involved in the quality analysis. Plant physiology, after myself, Dr. Kamurcu will present his research on this. And myself, I'm basically involved in molecular genetic parts of this story. Actually, our group is a little bit wider than the ones that I mentioned. Different disciplines also related to this field are involved in our group. And as I mentioned, abiotic stress is our basic target. And when I tell this, I mean actually boron and salinity. Boron, both deficiency and toxicity of boron are problems in my country and elsewhere. And salinity. Earlier it was thought that we lost huge amounts of soil every day to salinity, but I will start with our experience on boron research. Our group started this research during the late 90s in Central Anatolia in Turkey. Is known as the cereal storehouse of Turkey. Main crop is wheat. We found out that there is some 27% of this land boron deficiency and some 18% boron toxicity. We made this analysis for years with thousands of samples in our elemental analysis lab. Later on, when we discovered that there is such a boron problem in our central Anatolian soil, a nationwide survey started, and a much more saturated map is recently generated. We have a boron research institute in our country, and as you may see, there is a huge amount of boron deficiency in our land. But actually, there is also a toxicity problem. I will focus on the second part today. Just because Turkey is a boron-rich country, we hold about three-fourths of all boron resources in the world. So we can produce boron-enriched fertilizers, and that's what we have done. So there is a solution for deficiency, even if it's widespread. But when we consider toxicity, it is much more difficult. For years, we made trials in the fields using different crop plants, and we tried to understand the reaction against boron under deficient and toxic conditions. Later, we started doing more controlled environment studies in our greenhouses with different crop species. This is how boron toxicity affects a plant, a common bean. Here. We use different plant species, and later on we started also a much more controlled environment studies, which is under hydroponic conditions. Professor Kamurcu is involved in this research. We also started using wild varieties, wild species, close to wheat, of course, in general, to understand what's happening. We started looking for interaction with different elements and try to understand what the mechanism behind is. We found out that in an important crop like maize, there is a great variation in terms of reaction against this environment, toxic conditions. When we apply a certain high amount of boron, most of the hybrid varieties we used were not even able to germinate. But interestingly enough, there were some varieties that they perform pretty well, even at very high concentrations, like here. Another interest we had, maybe much more related with this meeting, we applied also salt in these experiments. But interestingly, when we apply directly salt, as you see, this high concentration disturbs all the plant. However, when we apply also certain amount of boron, we see that the very same plant survives. So there is a good relation. That was a start point for other experiments, but we also understood that there is a huge variation within species, but also between species, in the species level. So we started looking for new plants, basically in mining areas, different mining areas of Turkey. I would like to mention especially to this plant, this tiny grass. This is important for us, but there are also other plants. In certain areas, like the boron mining areas, we sometimes find only 1-2 species, like here. An important species there in this mining area is Gypsophila. Even if it is pretty good in terms of tolerance, our interest was on the other one, the Buccinella distance. I will explain this. This is related with our work with Gypsophila. It performs pretty well, even in very high concentration of boron. Why the other one? Because it is close to wheat, and we are in the wheat zone, and we cannot produce today enough wheat. Actually, coming from central Anatolia, wheat has been always a very important crop. Even during the Hittite period, even much more earlier time, during the start of civilization, some 8,000 to 10,000 years ago, Çatalhöyük is only 30 kilometers from the central part of Konya, and it is one of the few settlements on earth, first settlements. There are certain interesting things about this region, but to keep short, I don't want to go in any detail on this. Just to mention, we collected samples from different excavation areas, including the Çatalhöyük, in all over Turkey, dated some 6,000 to 8,000 years old. And we started determining their profile, what they were looking like. I mean,